So let's describe the organization of a skeletal muscle. We've already looked at the cellular structure for a skeletal muscle, but taking this same picture, but having the tricep cut in the image, let's imagine having that cross section and looking at the end of the tricep. Here we have something similar to the picture that we saw previously, looking at the end of the muscle. We have the tendon, we have blood vessels within the muscle, and so on. The various parts of the muscle here are the fascicle. The fascicle is actually a bundle of muscle fibers. Here we have the epimysium, just underneath the fascia that we were discussing before. It's another layer of connective tissue surrounding the muscle, primarily made of collagen. Here we have the perimysium. The perimysium is again connective tissue. It separates the muscle into compartments. Each compartment has a fascicle inside it, and it has neurons and blood vessels running through it that provide nerve and blood supply to the muscle. Around each of the muscle fibers, the cells, there is the endomysium, the connective tissue that wraps around each within the fascicle. If we were to actually look at a single muscle fiber, it would look something like this, except this is open in such a way that the cell membrane, called the sarcolemma, has been removed from the front of this image. This is so we can see the inside of the muscle fiber. Inside the muscle fiber, we have many myofibrils. Myofibrils are bundled within the muscle cell, and they are made up of two filaments, actin, which is thin, and myosin, which is thick. These make up the contractile units of the muscle cell, and we'll take a closer look at them in a moment. We have the sarcoplasmic reticulum. This is a tubular system found within the cell that helps to store calcium. As you can see here, the calcium within the pink area, the sarcoplasmic reticulum. Crossing the cells, we have the T-tubules. The T in this case stands for transverse, because the tubule goes across, like the transverse plane went across our body, it goes across the muscle fiber. Coming back to the myofibrils, let's look at this portion here of the myofibril. It's called a sarcomere. Running all up and down the myofibril are many sarcomere units, one after the other, going all the way along. If we zoomed in on the sarcomere, we would see this. I mentioned before that there is the thin protein filament, actin, and the thick protein filament, myosin. In this image, you can see that the myosin also has protein heads sticking off of it. At either end of the sarcomere, we have Z lines, which represents the end of one sarcomere and the beginning of another. During muscular contraction, the myosin heads attach to the actin and form a cross bridge, and then draw the actin in towards the center of the sarcomere. Overall, the sarcomere gets shorter, and because we have many of these repeating all along those myofibrils, all along the muscle ultimately contracts. This is related to the contractility of the muscular system that we mentioned earlier in the unit. We'll go more in depth into that in a moment here, but let's focus now on the excitability characteristic of muscle that we discussed as well, by explaining the relationship between the muscle and nervous systems. Each skeletal muscle fiber is connected to a nerve cell. That nerve cell, the motor neuron, that comes out of the motor nerve that comes from the spinal cord, attaches to multiple muscle fibers, and that overall is the motor unit, a single motor neuron and the muscle fibers that it attaches to. This allows for communication down from the central nervous system to the periphery. In order for a muscle cell to contract, it must receive a stimulus from a motor neuron. To go a little bit deeper into that then, let's explain the relationship between these various terms and portions of this connection. Here we have the end of a motor neuron and the surface of a muscle cell. Connecting the two is the neuromuscular junction, the end of a motor neuron and the motor end plate of a muscle fiber, the portion of the muscle fiber where the motor neuron attaches. The synapse, or the synaptic cleft, is a narrow space between the end of the motor neuron and the muscle fiber. The electrical signal that comes down the motor neuron must get across the space to the muscle in order to produce a contraction. However, the electrical signal cannot be passed directly to the muscle. The motor neuron transmits that electrical signal down from the central nervous system, and when it arrives at the end of the motor neuron, the electrical signal stimulates the synaptic vesicles, the little circles here with the neurotransmitter in them, to move to the surface and then release into the synaptic cleft. The neurotransmitter then diffuses across the synaptic cleft over to the receptors on the muscle cell. When the neurotransmitter attaches to the receptor sites on the muscle, it allows the signal to then move along the muscle. Thus, the nerve impulse has been transferred to the muscle via this chemical signal. So, what is the body's main neurotransmitter? Here we can see the neurotransmitter has ACH, but this is just a short form for acetylcholine, which is the main neurotransmitter of the body, bringing the signal from the motor neuron to the muscle cell. Once that signal goes over to the receptors found on the muscle cell, how does the muscle contract and relax? The electrical signal, the nerve impulse, travels down the nerve to the terminal and causes the release of the neurotransmitter acetylcholine, as we mentioned before. The acetylcholine diffuses across the neuromuscular junction, the synaptic cleft, and binds to the receptor sites. The stimulation of the receptor sites causes an electrical impulse to form in the muscle membrane. The electrical impulse travels along the muscle membrane and penetrates deep into the muscle through the T-tubule system that we saw in this image earlier. As that signal travels down the T-tubule, the impulse stimulates the sarcoplasmic reticulum to release calcium into the sarcomere area. The calcium allows for actin, myosin, and ATP to interact, causing cross-bridge formation and muscle contraction. The process continues as long as calcium is available to the actin and myosin. So if we were to zoom in on this area here, we would see this. At the bottom of the image, we have the myosin heads. At the top, we have the actin. On the actin, we have myosin binding sites, the spheres that are covered by the tropomyosin, another protein. Attached to the tropomyosin, we have troponin. When the calcium is released from the sarcoplasmic reticulum, it will bind to this area of the troponin. When it does so, you can see here that it changes the shape of the tropomyosin enough that the tropomyosin moves off of the myosin binding sites found on the actin. This allows the myosin heads to attach to the myosin binding sites and then pull on the actin, drawing it in. This is related to the bottom right-hand picture of the contracted sarcomere, where all of those myosin heads have attached, forming cross bridges to the actin, and then pulled them towards the center of the sarcomere, making the sarcomere shorter. Once this process finishes, muscle relaxation occurs when the calcium is pumped back into the sarcoplasmic reticulum away from the actin and myosin. When calcium moves in this way, the actin and myosin cannot interact any longer, and the muscle relaxes. Overall, that is the process of muscle contraction and relaxation. But to zoom in a little bit closer on the myosin and the actin, we can also look at the role that ATP plays in the contraction. When ATP binds to the myosin head, it removes the myosin head from the myosin binding site on the actin. Then calcium comes in, as we saw before, and binds to the troponin that moves the tropomyosin out of the way of the myosin binding sites on the actin. The ATP is broken down into ADP and a phosphate, which gives off energy, allowing the myosin head to attach to the myosin binding site and then to actually move. What happens as that movement occurs is the ADP and the phosphate are released from the myosin head. And then to detach the myosin head from the myosin binding site on the actin, we repeat this process. A new ATP comes in, attaches to the myosin head, removing it from the myosin binding site. This goes along with what we already discussed. The calcium at this point is pumped back into the sarcoplasmic reticulum. Here we have another image of what we've been discussing in terms of a motor neuron attaching to the motor end plate of a muscle cell. We have synaptic vesicles containing acetylcholine, and then receptor sites on the motor end plate for acetylcholine. But we want to use this to discuss how the threshold stimulus and all or none principle relate to muscle contraction. The minimal strength needed to cause a muscle contraction is called the threshold stimulus. We actually need to have enough acetylcholine come across from the neuron to the motor end plate in order to stimulate those receptors to cause the muscle to contract. A muscle fiber will remain inactive until the stimulus is strong enough to cause the threshold stimulus. Once the threshold stimulus is reached, the response will be that the muscle cell will contract to its fullest extent. All of the sarcomeres that you see inside that muscle fiber will contract. There is no such thing as a partial contraction of the muscle fiber. It is an all or none event. So the threshold stimulus is the minimal strength of stimulus needed to cause the muscle to contract, and the all or none principle is that once that muscle fiber starts to contract, it contracts completely. One of the key things to note here is that we're talking about the muscle fiber, the muscle cell, not the muscle as a whole. So it wouldn't work to say that the entire muscle must contract once it starts to contract. And this is because when a muscle contracts, depending on what we're actually moving or using our muscle for, we may only have some of the muscle fibers actually being signaled to contract. Let's take a closer look at that. Here, we have a number of neurons coming out of the spinal cord and attaching to various muscle fibers within a muscle. So we have 3 motor units here, represented by the 3 colors shown in the grid. If we were to lift something light, like a pencil, this is not a situation where our muscle fibers throughout our muscle are only partially contracting, because that does not happen. What's going on when we lift something light, like a pencil, is that we only stimulate so many motor units. So in this case, representing it by 1 being stimulated and stimulating 3 muscle cells that fully contract, but is only producing the small amount of force that is required to lift a pencil. What if we needed to lift something heavier? What happens is we actually send signals along more motor units. This process is called motor unit recruitment. We recruit more motor units to produce enough force to lift this heavier weight. In this case, all 3 motor units are being signaled, which means many more cells are actually contracting to lift the heavier weight. One thing to note here is that muscles with fine motor control, like in our eyes and fingers, have many small motor units. Muscles with less precise movements have larger motor units. So what are the effects of exercise on muscles and the overall body? Hypertrophy is an increase in size due to use. If we break down this term, hyper means increase. Trophy is size. So as you use your muscles, you exercise, your muscles increase in size. Here we have an inactive muscle, and after exercise, the muscle overall gets bigger. This is not always positive, as in our example here. In cardiac muscle, hypertrophy is something that you don't want to have happen. As the cardiac muscle increases in size, there is less space in the heart for the blood to get into, therefore having a negative result. Atrophy is a decrease in size due to lack of use, so the opposite of the hypertrophy that we just saw.